So following on from our discussion about transport of oxygen in the blood, I want to talk about carbon monoxide now. And even if you're not studying physiology, it's useful to know about carbon monoxide because it's a very dangerous chemical and it can be produced when somebody's not using a stove correctly or if there's a fire somewhere nearby. And this is its chemical structure. So it's a single carbon atom with a triple bond to an oxygen atom. One of the bonds is dative covalent. And this is what happens when it comes into proximity with hemoglobin. So it binds to hemoglobin to form carboxyhemoglobin. And this is not good because the affinity of carbon monoxide is 240 times higher than the affinity of oxygen for hemoglobin. So in other words, hemoglobin binds the same amount of carbon monoxide as oxygen when the partial pressure of carbon monoxide is 240 times less. So you only need a small amount of carbon monoxide in the air to decrease the binding to oxygen, and it binds hemoglobin in place of oxygen so that hemoglobin can't bind oxygen, so you can't get enough oxygen around your body. So it interferes with oxygen transport in the blood. Now to illustrate the effects of this, we can plot a graph of the partial pressure of oxygen in millimeters of mercury against the total oxygen content of the blood in milliliters per 100 milliliters of blood, which includes the oxygen bound to hemoglobin and the oxygen dissolved in the plasma. So this first curve is the normal blood, where there's no carbon monoxide around and the oxygen content of the air is around 21%. So the O2 content is normal, it's around 20 milliliters per 100 milliliters of blood at a partial pressure of 100. Now compare this with the second curve, which is when there's just 0.1% of carbon monoxide in the air, so a very small amount of carbon monoxide, and it's almost negligible. Well, not in this case, but it's very small, and the oxygen content of the air is still 21%. So now we can see that the oxygen content of the blood has decreased significantly by 60%. 60% of the hemoglobin in the blood now is carboxyhemoglobin. And you can imagine the implications that it has for normal function, so you can't get enough oxygen to your tissues and you get hypoxia. Another thing that carbon monoxide does is it shifts the Bohr curve in the tissues to the left, so it increases the affinity of hemoglobin for the oxygen in the remaining little 40% that is still bound to hemoglobin, and that means it's much more difficult to unload oxygen from the remaining hemoglobin that you have, and this results in hypoxia again. Why is this so dangerous? So the partial pressure is normal, but the total oxygen content of the blood dramatically decreases when you have carbon monoxide poisoning. And normal arterial partial pressure impairs the chemoreceptor mechanism. So the chemoreceptors are the detectors for partial pressure of oxygen, and even if the O2 content is low in the blood, they won't detect it unless the partial pressure is also affected. So basically, you don't have any negative feedback mechanisms to help with this situation. Also, there's no physical signs of hypoxia, and there's no symptoms or anything. There's no cyanosis, which is the skin turning blue, because blood stays red when bound to carbon monoxide. It's also odourless, colourless, and non-irritating. This means that you have to be exposed to lethal amounts of carbon dioxide before you start to notice, and by then it's often too late. And your blood has decreased oxygen content to anoxic levels with no outward signs. This is why it's important to have carbon monoxide alarms, just like smoke alarms in your home, and treatment involves pure oxygen therapy, which competes off the carbon monoxide bound to hemoglobin. Now let's have a look at how carbon dioxide is transported in the blood. So we've talked about oxygen transport, and carbon dioxide transport is slightly more complicated. There's three ways mainly that it can be transported. So firstly, dissolved, secondly, bicarbonate, which is most of the carbon dioxide in the blood, and thirdly, as carbamino compounds. So this diagram here, hopefully you can see it clearly, it shows quite well how the carbon dioxide is transported. So we're in the tissues, and the tissues are in an intimate relationship with the plasma and erythrocytes. So the carbon dioxide is in a high concentration in the tissues, which have just respired, and it diffuses down its concentration gradient into plasma and then into the erythrocytes, the red blood cells. And as it diffuses down its concentration gradient, there's various mechanisms which allow it to be stored and transported in these mediums. And this part is about oxygen transport. So we'll have a look at these three main methods of transport. So firstly, carbon dioxide can be dissolved in the plasma. So unlike oxygen, substantial amounts, about 5 to 10% of the total carbon dioxide content of the dissolved carbon dioxide in the blood is transported as dissolved carbon dioxide in the plasma. So you can see here in the plasma it's dissolved, and there's a tiny bit dissolved in the red blood cells as well, 5 to 10%. And this is about 28 millilitres of carbon dioxide per litre of plasma in arterial blood. Now the carbon dioxide solubility coefficient in plasma is much higher than that of oxygen. It's about 0.7 millilitres of carbon dioxide per litre of plasma per millimetre of mercury, which is 23 times that of oxygen. And Henry's law states that concentration equals partial pressure times solubility. Therefore, at a partial pressure of carbon dioxide of 46 millimetres of mercury, which is standard for systemic venous blood, we have 46 times 0.7 equals to 32 millilitres of carbon dioxide per litre of plasma. That's just an average figure. Now, the second way that carbon dioxide can be transported in the blood is as bicarbonate, which has the formula HCO3-. And the majority of carbon dioxide in plasma and the red blood cells is hydrated. So what happens when water inside the plasma and the red blood cells reacts with the carbon dioxide that's diffused down its concentration gradient is this. So carbon dioxide plus water reacts reversibly to form carbonic acid, H2CO3. And in the red blood cells, this is a much faster process because an enzyme called carbonic anhydrase is present, which catalyzes this process, but it also happens to a smaller degree, slower in the plasma. Now, carbonic acid has a low pKa, so it's a strong acid, which means that it releases hydrogen ions more easily, and the dissociation constant is about 6.1. So the lower the dissociation constant, the stronger the acid, and the more protons are generated at a given pH. So at pH 7, physiological pH, there's much more protons generated by carbonic acid, so it dissociates pretty easily. And we can see on this diagram that in the plasma there's around 90% of the carbon dioxide is transported as carbonic acid, and this is the case in erythrocytes as well. So the reaction between water and carbon dioxide in the red blood cells is much faster because there's carbonic anhydrase present, but the carbonic acid that's formed will dissociate, and then the HCO3- will diffuse back down its concentration gradient into the plasma. And the last way that carbon dioxide can be transported in the blood is bound to proteins as carbamino compounds. So proteins reversibly bind carbon dioxide to amine groups. Plasma is around 7% protein, so there's many amine groups present for the carbon dioxide to bind to. Another equation here, so RNH2 representing a protein with an amino group plus carbon dioxide molecule reversibly reacts to form RNHCOOH, which is a carboxylic acid. And then this dissociates depending on the dissociation constant, which is less than 6, so it's pretty acidic. It dissociates into an anion and an H+ ion, so it releases H+ pretty easily. And to stop the blood becoming too acidic from this process, the large pH change has to be buffered by plasma proteins. So we can see here that in the plasma and in erythrocytes there are carbamino compounds present, so the proteins have combined with carbon dioxide to form these carbamino compounds, and in the plasma it could be any proteins, so 5 to 10% is transported this way. In red blood cells it's mainly hemoglobin is the protein that carbon dioxide binds to, but they're still the same carbamino compounds. And red blood cells have around 30% hemoglobin, so there's plenty of hemoglobin for the carbon dioxide to bind to form carbamino compounds. So those are the three main ways that carbon dioxide is transported. A few other things now. So we said that the bicarbonate is how most of the carbon dioxide is transported. It has to diffuse down its concentration gradient from the erythrocytes into the plasma. So surely then the red blood cells would become too acidic. Well, the charge needs to balance out, and this is what Hamburger's phenomena is for. So otherwise known as chloride shift. When the bicarbonate ions leave the erythrocytes and enter the plasma, down the concentration gradient, chloride moves in, so it balances out the loss of negative charge there. And this is a pretty rapid exchange because the red blood cell has very high anion permeability. All the extra chloride ions going in increases the intracellular osmolarity of the red blood cell, and that also causes water to go in, so the red blood cell swells. And now we have a look at this bit, which I haven't touched on yet. So in the erythrocytes, the carbon dioxide combines with water, dissociates, and after the bicarbonate diffuses, the protons are left. Now, to stop the blood becoming too acidic, the protons combine with hemoglobin. So they form hemoglobinic acid, or HHV. And the way they do this is by the imidazole groups of the histidine amino acids of the alpha and beta polypeptide chains on the hemoglobin protein. And the hemoglobin protein has a pKa of around 7, so they're pretty good buffers. Now, you'll probably see that this is a familiar phenomenon because as the H plus displaces the oxygen, it's then released. So this is actually the principle of the Bohr effect. The Bohr effect in tissues, to remind you, is when there's increasing carbon dioxide, so decreased pH and increased H plus, then more will combine with water and produce more H plus ions. So increased carbon dioxide essentially is the same as decreased pH. Now these extra H plus ions will combine with more hemoglobin, and H plus has a high affinity for hemoglobin than oxygen, so it will displace the oxygen and allow it to diffuse down its concentration gradient into the tissues, which explains why there's a right shift. So there's a decrease in affinity for hemoglobin when there's a lot of carbon dioxide. Now we move on to the Haldane effect, which I like to call the reverse Bohr effect. So after I explain it, you'll probably be able to judge how accurate that is. I think it's pretty accurate anyway. So the Haldane effect states that at an increased partial pressure of oxygen, the blood carbon dioxide content is decreased. So there's increased carbon dioxide content in deoxygenated blood, which is saying the reverse of this basically, than oxygenated blood, because the H plus dissociation constant for hemoglobin is dynamic, so it can change depending on the concentration of oxygen. And this results in the fact that deoxygenated hemoglobin is a weaker acid, so it binds more protons at a given pH. For example, at a low partial pressure of oxygen, for example in the tissues, it might not quite be 0, but it will be low. So when it's low, you can see that the carbon dioxide content is higher at a given partial pressure of carbon dioxide in the blood. And this is because the hemoglobinic acid concentration has increased, and that has also increased the bicarbonate production. So more carbamino compounds are formed as well. And if there's more bicarbonate, then there's more CO2, because that's the way that it's stored, remember. Now, how are the movement of gases maintained? So maintaining the carbon dioxide diffusion into the red blood cell is pretty important, because we need that gradient for it to be able to exit the tissues and into the red blood cell. So there's continued bicarbonate efflux from the tissues, and this is as a result of the production of carbamino compounds and H plus buffering by hemoglobin. And Haldane effect also increases the concentration of bicarbonate at a low partial pressure of oxygen in the blood. Acid-base control during respiration. So we're going to talk about how the pH of blood is calculated and some key equations relating to acid-base control. So this is an equation we've seen before, dissociation of carbonic acid. Now if you didn't know already, pH is equal to minus log of the concentration of hydrogen ions. So when there's a low pH, there'll be more hydrogen ions and vice versa. Now the Henderson-Hasselbalch equation tells us how to calculate pH from the dissociation constant and the concentrations of the anion and the acid. So pH equals pKa plus log of concentration of bicarbonate in the blood over concentration of carbon dioxide. Now carbon dioxide obeys Henry's law, so the concentration of carbon dioxide equals the partial pressure times the solubility, which is 0.03. The pH of the blood is determined by the amount of carbon dioxide in solution and the carbonic acid dissociation. So if we combine Henry's law and the Henderson-Hasselbalch equation, we can find that pH is equal to pKa plus log of the concentration of bicarbonate ions over 0.03 times partial pressure of carbon dioxide. Now we can use this to work out the pH of blood. So what is the pH of blood if we know that its pKa, dissociation constant, is 6.1, and normal arterial blood concentration of bicarbonate ions is 24 millimoles per litre? Well, we plug into the numbers, so pH equals 6.1, the pKa, plus log of 24, normal arterial blood concentration of bicarbonate, over 0.03 times 40, which we know is the normal partial pressure of carbon dioxide in the blood. And that equals 6.1 plus log 20, so 20 is the ratio of bicarbonate to carbon dioxide in the blood, which equals 6.1 plus 1.3 equals 7.4. Now, if the ratio of bicarbonate to carbon dioxide remains 20, then the pH remains constant at 7.4, and you might think that this is a poor buffer system if you know something about buffers, because we want the pKa to be closer to the pH for a good buffer to work, to keep the pH close to 7.4 as possible. However, 6.1 is pretty far from the pH that we're trying to buffer, but it's okay because the carbon dioxide bicarbonate buffer system is an open buffer, which means that the concentration of carbon dioxide can be easily modulated by the lungs. So remember the alveolar ventilation equation and that relationship, we increase our ventilation to decrease the concentration of carbon dioxide, and the lungs excrete around 10,000 MEq of carbon dioxide per day. The kidneys also contribute to bicarbonate control, so these two factors are controlled pretty well and don't need a very strong intrinsic buffer to maintain the pH of blood. The last thing we're going to look at is a Davenport diagram, and this easily shows the relationship between pH, the partial pressure of carbon dioxide, and the concentration of bicarbonate in the plasma. So if we look at the bottom here, we know that 7.4 is the normal pH of blood, and any deviations from that are pretty dangerous. So if we go down to 7.1, that would definitely be very bad. That's acidosis. And anything above that is alkalosis. So the body has to keep pH relatively constant within a very narrow range. And how does it do this for varying levels of partial pressure of carbon dioxide? So at 20, we'll see that it's pretty low partial pressure of carbon dioxide, so we're beginning to get alkalosis, and as it increases, we get acidosis. So the optimal place would be in the middle here, and there's various things that can happen, and this diagram shows how they're compensated. So the first thing that can happen is metabolic alkalosis. So the pH of the blood increases because the plasma bicarbonate concentration increases, and this can be due to ingesting alkali or vomiting, which can also cause alkalosis, and this is corrected for by respiratory compensation. You can breathe less, but it doesn't always happen. Another type of alkalosis is respiratory alkalosis. So the plasma bicarbonate levels don't really change that much, but the partial pressure of carbon dioxide has changed drastically. So for example, if you're hyperventilating, then you'll decrease your partial pressure of carbon dioxide by quite a bit, and that will cause respiratory alkalosis. So it's important to be able to distinguish between metabolic alkalosis and respiratory alkalosis. Metabolic alkalosis has an increased plasma bicarbonate concentration, whereas respiratory alkalosis does not. It's caused by the PCO2. And here we have renal compensation. So respiratory alkalosis is corrected for to a certain degree by the kidneys excreting more bicarbonate. In the other direction, we can have respiratory acidosis, which is when we don't breathe enough, essentially, and carbon dioxide starts building up in the blood, or hypoventilation, and you can see that the partial pressure of carbon dioxide has increased. Now, this is also compensated for by the kidneys, and it increases the level of bicarbonate in blood. The other type of acidosis is metabolic acidosis. So if your blood acids increase for some reason, then you can get metabolic acidosis where the plasma bicarbonate decreases significantly, but the PCO2 remains the same. And this is compensated for by the lungs. So we hyperventilate, we breathe more and more frequently and more deeply to increase our alveolar ventilation, and we can increase the partial pressure of carbon dioxide in the blood and in the alveoli. So the key point is that the body wants to make sure that the pH of the blood is as close to 7.4 as possible. Full compensation is unlikely to happen in practice, but the body tries as hard as it can to maintain this pH because acidosis and alkalosis has very negative effects, and we need to keep it within a narrow range.